Hi guys, in this video I'm going to walk you through the process of a virus infecting a host cell, and so I'm going to use lots of vocabulary that is relevant and you should do the same. So first and foremost, a virus is not technically alive because it can't reproduce or metabolize on its own. It actually needs a host cell in order to do so. And so this is a very basic structure of what is called a virion. Essentially one individual viral entity. And so what I want to point out first is that the virus is actually surrounded by a phospholipid bilayer. So it's called a membranous envelope. Sometimes they have a couple membranous envelopes, and you see that it is the exact same structure as a human cell. Okay, and so every time you see this little black line here, that represents a phospholipid bilayer. Embedded within that phospholipid bilayer are receptors, so proteins that are coded for in a very specific way. They have a very particular shape, and that's actually how they're going to infect and actually get inside our cells. Okay. Within the membranous envelope, oftentimes virions have a protein capsid, right, also coded for by the genetic material of the virion. And so the virion's genetic material is super short compared to living organisms. It can be single-stranded, double-stranded, and it can be DNA or it can be RNA. Okay, so they have a lot more options than living organisms do. Also, the single-stranded, for example, can be either a positive or a negative. So sometimes the viruses actually have to make a copy or have the host cell make a copy to go from negative to the positive that actually codes for its proteins. Okay, and so the example virus that I'm going to talk to you about today is a single-stranded RNA positive, which means that there's only one strand. It is RNA, so it can be immediately translated into a protein, and of course it is positive. And so this example is just like COVID-19. So we're going to continue with that example of COVID-19. The receptors, the proteins embedded within the membrane of, in this example, the COVID-19 virion, have a complementary shape that matches a receptor, a type of receptors that's already in a lot of our cells, particularly the cells that line our respiratory passages. And this receptor is called an ACE2 receptor. This is actually involved in your blood pressure regulatory pathway. And so it's really important, and these receptors are all over the place because regulating blood pressure is, of course, super important. And so these proteins on the surface of COVID-19 actually bind to the receptor that's already in our cells, and we need this as a good receptor, but what that's going to do is it's going to stimulate a process called receptor-mediated endocytosis. And so as we can see, the phospholipid bilayer of our host cell would then start to invaginate. It starts to form a little depression, and ultimately is going to form a little vesicle. And this vesicle is going to be pinched off and enter into the cell, and now we call it an endosome. Okay, so all of this is happening in the cytoplasm. Once this endosome, this little vesicle, is inside the host cell, what's going to happen is essentially denaturation of all of those protective coatings on the outside of the virion. So here we can see hydrogen ions are being actually pumped into this little vesicle. As we know, this causes denaturation, so the pH is going to go way down. And ultimately, all of those other things are going to unravel, therefore freeing up that RNA. Okay, so now we have RNA. COVID-19 has less than 10 genes, but obviously they're very, very specifically adapted for their purpose. Okay, so we have mRNA. Here's the beginning, right, and here's the end with the poly A tail. As we know, there are free ribosomes that are circulating through the cytoplasm. These ribosomes, so the host cell, our ribosomes are going to start reading this mRNA from the 5' end, just like it would with human mRNA. It's going to find the start codon AUG, call in some tRNAs, and ultimately start synthesizing this protein. So this is the translation process that would happen with our normal cells. Okay, there are indicators within this mRNA that this process needs to be completed in the rough endoplasmic reticulum, the RER. And so what we can see here is the ribosome, the mRNA, and ultimately the little proteins that are being made are ultimately going to dock on the surface of the RER, and then translation is going to continue. And so ultimately we're left with a string of amino acids that is embedded within the RER membrane, right? And this is happening for all of the genes in the viral genome. And so as we know, the RER is going to process proteins into their secondary and tertiary structure and also add those carbohydrate hedges. And so by the time the RER is done with these proteins, they're going to look like those same receptors which helped the virion actually invade the cell in the first place. Okay. Also, there are other genes, of course, not just these receptor genes, but that protein capsid. So here, the protein capsid also being processed by the RER, and a few other genes as well. Okay, and so these proteins, right, so now these are viral proteins made by the human host cell. They are going to be packaged up into little vesicles, either embedded within the membrane or just shoved inside this little vesicle here. And as we know, they are carried across the cell to the Golgi apparatus. Okay, and that happens via microtubules and carrying by molecular motors. Okay, and so as we know, the Golgi's job is to sort proteins. And so ultimately, by the time the Golgi is finished, what we have is a new virion. So here's the protein capsid packaged up with the RNA genome. And the membrane, right, which happens to be this initial RER membrane, right? So the virus didn't make it itself, we made that. So this is the phospholipid bilayer that initially was part of the RER, got passed through the Golgi, and now this virus is ready to leave the cell to go infect another cell. Okay, so this process is called exocytosis. Essentially, the transport vesicle, which held onto this virion, is going to fuse with the membrane of the surface of the cell and ultimately is going to release it out into the world. And so, or out into the body, and then the body would then release it out into the world. And so this, of course, is happening over and over and over again. This single host cell is producing thousands of these individual virions, each capable of infecting a new cell. There are other viruses that actually have the ability to take their viral genome and insert it into our genome. And so generally, those viruses do not kill their host cells. Instead, they are going to keep employing that host cell to keep on pumping out more and more and more viruses. And you never really get rid of those because the viral genes are in your own genome. So that is the process of infection by a virus, specifically because of this right here. This is COVID-19. Thanks for watching.